In this video, we're going to look at protein structure and see the different levels of protein structure and what holds it together. There are 4 levels of protein structure: primary, secondary, tertiary, and quaternary. We're going to look at each of these and look at what those structures are, and then what are the forces or interactions that hold them together. As we go through all of these, the important thing that you need to remember from this, the takeaway from this, is you need to know 2 things for each of these levels of structure. You need to know what they are, and the second thing is, what are the forces or interactions that hold it together. So those are the 2 main takeaways from each of the levels of structure. So starting off, we have primary structure. Primary structure is simply the amino acid sequence. So simply just the order the amino acids are bonded together. The force that holds those amino acids together is a peptide bond. And in my video on amino acids and peptides, I showed you what that peptide bond looks like. So that is the thing that holds it together, holds the amino acids together. And the primary structure is simply the order that the amino acids are in. The secondary structure is how that primary structure, how that amino acid chain, that polypeptide chain, folds together. And if it folds into a regular structure, that's our secondary structure. And those regular structures that it could be are the alpha helix or the beta pleated sheet. Alpha helix are coils. So it's in my picture here. It is this green coiled section. And so they do look like that. They are just amino acid chain, that polypeptide chain that just coils around itself. And it's held together through hydrogen bonds of the backbone region of that polypeptide chain. The other regular shape that it can have for secondary structure is a beta sheet. And this drawing right here for both the alpha helix and beta pleated sheet is what we call a ribbon model. It's just a type of illustration to show the different structures of proteins. And beta sheets in this ribbon model are often illustrated as arrows going in different directions. And so the polypeptide chain is the gray. The beta sheet part are the arrows. And it's oftentimes called a beta pleated sheet. How the structure is, it almost looks like it's a sheet of paper that has been folded back and forth and kind of opened back up, or like the inside part of a corrugated cardboard piece where you have up and down sections of that. But you have this regular shape going back and forth, and they are also held together through hydrogen bonds in the conserved backbone region of the chain. Now we come to a tertiary structure, and this tertiary structure is how your overall protein is folded. Most proteins are globular in nature, and so proteins serve a function biologically, and they serve that function when they're in their active form. And so in this is what the tertiary structure is, kind of the active form of your protein, how it folds up biologically to be able to perform whatever its job is. Now, tertiary structures are held together by a lot of different types of interactions, but they are held together between interactions of the R groups, those side chains, the things that differ from one amino acid to the other. And those interactions involve or include hydrogen bonds. So it can hydrogen bond between a hydrogen in one side chain and an oxygen in another side chain. You can have hydrophobic interactions where you have those nonpolar amino acid side chains facing each other. Most of them are going to be folded towards the inside of your molecule because remember, this is in biological conditions. Your cells are aqueous, and so those hydrophobic side chains fold towards the inside of these globular molecules, and it keeps water outside of it, and they have those interactions between hydrophobic or nonpolar amino acid side chains. Hydrophilic interactions, so these are going to be some interactions with water, side chains on the outside of your globular protein here that are interacting with water on the outside. Salt bridges, this can occur between an acidic and a basic side chain where you have opposite charges, and so they are attracting because opposite charges attract, and so it forms a salt bridge, an interaction that holds those two parts of the molecule together. And then finally, disulfide bonds, and this would be between two cysteine amino acids. Remember, in a cysteine, you have a sulfahydryl group, or a sulfur bonding to a hydrogen group. Well, those hydrogens can be reduced, and you can form a bond between one sulfur and one cysteine and a sulfur and another cysteine. That's what the disulfide bond is. All of these interactions hold this three-dimensional shape together. And you can see in this picture of the tertiary structure, you can see the different secondary structures, a couple alpha helical regions. You can see the ribbons, the arrows here forming some beta sheet regions. And so, but it's how all of those things interact together, and the three-dimensional shape that it has is its tertiary structure. Some proteins in their active shape contain more than one polypeptide, more than one amino acid chain in its final form. And so if a protein needs more than one separate amino acid chain, it would have a quaternary structure. And that quaternary structure is just how those individual polypeptide chains that have their own primary structure, own secondary structure, and folded into its own tertiary structure. So how those separate tertiary structured chains, subunits, come together and stick together in an overall unit. And so that overall unit would be the active form of that protein if it has more than one chain that makes it up. And that is the quaternary structure, just how it all fits together. So quaternary structure is made from more than one subunit, and they are held together by those same interactions that hold the tertiary together. So it's held together by hydrogen bonds, hydrophobic interactions, hydrophilic interactions, salt bridges, and disulfide bonds. Those are the forces that hold quaternary structure together. So those are the four structures that are found in proteins. And again, you need to remember those four structures, what they are, and then what forces hold them together.